Alrighty, can you hear me? Awesome. Thank you so much for the opportunity. I'm really excited about this. So I hope you like it and enjoy it. So hello everybody. Today I'm going to present part of our work in the Almodovar's lab, focus on drug delivery that we have published. My name is Katherine Miranda from the University of Arkansas. So I'll start talking a little bit about myself. As you mentioned, currently finished my first semester as a grad student. I'm a PhD student in biomedical engineering at the University of Arkansas. I'm from Panama. I graduated this May, bachelor in biomedical engineering as well here at the University of Arkansas. So during my bachelor's, I had the opportunity to work in the polymeric biomaterials lab under the mentoring of Dr. Jorge Almodovar from the chemical engineering department. So in his group, I have the fortune to work with two different projects. The first one was related to the cell manufacturing of human mesenchymal stem cells, and the last one related with the microneedle patch for drug delivery. This is part of the presentation of our website there. Some of the currently research that we are doing right now in biomimetic surface, regeneration of damaged neural tissue, and the main one, the one that I'm going to be talking about today. As I mentioned, I started working as an undergrad. I had the fortune to work with an awesome grad student. He already graduated. He's working right now in the University of Texas at Austin, Dr. David Castilla. So I'm going to be talking about this awesome project that we work together. So I'll start with a little bit of overview about what polymers are. So as you know, polymers have demonstrated being useful for the fabrication of biomaterials with excellent properties capable of replicating specific characteristic functions or mechanisms in the human body. Polymeric systems have facilitated the promotion of processes such as cell addition, proliferation, differentiation, and tissue regeneration. In addition, they have been abundantly used to prepare devices for drug delivery applications. Some examples of polymers are chitosan, the main one that we are going to be talking about today, hyaluronic acid, heparin, collagen, and there is many more. So polymeric biomaterials have been elaborated in several presentations, such as hydrogel, sponges, nanofibers, or in a multi-layers by using different techniques, such as layer by layer, spin coating, electrospray, and electrospinning. And all of these different characteristics have promoted the development of biopolymeric materials. Polymers can be classified based on their origin, structure, molecular forces, and the polymerization main existing mechanism. Today, our project will be based on the use of those ones that are from natural origin or natural polymers. So, as I mentioned, we are going to be focused on this, also this polymer, the chitosan. Chitosan is considered as one of the most promising biomaterials of the 21st century on account of its versatile nature, excellent biodegradability, biocompatibility, antimicrobial activity, and no toxicity, and wide applications. Chitosan is derived from the chitin, a second most abundantly available natural polymer after cellulose. The chitin is naturally found in the exoskeleton of shellfish, such as crabs, shrimps, and the excellent membrane of fungi, yeast, and other microorganisms. Chitin is not soluble in dilute acid, whereas chitosan is soluble in dilute acid. So this project is involved in animal welfare. Animal welfare describes the relationship that people have with animals, specifically related to the duty of guarantee that the animals under their care are treated compassionately and responsibly. Countries such as Canada, the US, and Australia have applying demonstrated interest in farm animal welfare, reporting their concerns during practices such as castration, dehorning, and tail duckling in cattle. The situation is that these practices are painful, but analgesia is rarely provided. Hence, many livestock animals are not handled daily. Countries such as US and Canada have been rigorously working on the well-being of livestock animals. The AVMA, which is the American Veterinary Medical Association, has been encouraging the use of pain relief during routine practices such as castration and dehorning in cattle. So, few pain medications exist commercially that are Food and Drug Administration, FDA approved for use in cattle. So some of the drugs have been used for that, for example, flunixin, but this drug has a degradation time of 6.2 hours, and it is necessary to dosage every 12 or 24 hours for about 2 weeks. So can you imagine if you have to treat around 1,000 cattle at the same time? It will be like a really hard work to do. So another drug that has been used is meloxicam, which has a degradation time of 28 hours. But this drug has also a disadvantage that is only available in table form, which is not available for livestock administration. So we need to develop a suitable way to control drug delivery of these pain medications. So the pharmaceutical field has used the molding technique to prepare membranes with different morphological characteristics. This is to present ways to deliver drugs such as tablets, capsules, and a sophisticated system like the microneedle patches, which is a platform composed of needles uniformly organized and designed to painless penetrate the skin. There is different type of microneedle patches, the solid microneedles, core microneedles, the solid microneedles, and the hollow microneedles. Our project will be focused on those ones that are dissolving microneedles. So therefore, our main goal is to engineer and test a chitosan-based polymeric microneedle patch for the transdermal delivery of meloxicam as a pain management approach for use in cattle. So our design consists of preparing a patch with 8 by 8 millimeters. In this area, there are 225 microneedles with 300 micrometers of base and 600 micrometers of height. The pitch was 250 micrometers. So to understand how the transdermal delivery happens, let's see the following figure. So we have this microneedle with hundreds of micrometers long, which is sufficient to cross the skin permeability area, the stratocorneum, but is short enough to avoid causing pain. The drug is directly placed in the epidermis or epidermis layer, which then goes through the blood vessels into the systematic circulation and show a therapeutic response on reaching the size of action, as you can see here. So to prepare the microneedle patch, we prepared 2 different solutions. The first one was one consisting of preparing chitosan solution by dissolving chitosan powder in acetic acid in water at a final concentration of 10%. The other solution consisting of adding and mixing 50 milligrams of meloxicam and 1 millimeter of chitosan solution. Then we have the we start adding the chitosan meloxicam solution, a mole of PDMS, approximately 5 milligrams of this solution. After that, we introduced the mole into a tube and it was under centrifugation for around 30 minutes. This resulted in the first layer. The next step was repeating 4 times step 1 and 2. Step 5 consisted of obtaining the second layer by adding chitosan solution. Step 6 was just drying the patch in the oven under these conditions. And finally, we were able to take off the patch. So some of the study that we performed in this project, we did a full characterization of the microneedle patch. We characterized the topography of the patch, the chemical composition, the mechanical properties. Also, we did an in vitro study to characterize the capacity to penetrate the skin. For the results that we got for each study, we have the following. For the physical characterization, we use the ACM. Figure A shows how the chitosan microneedle patch looks. Here we can see 1 microneedles, and figures B, C, and D show how the chitosan meloxicam patch looks by varying the concentration of the acetic acid during the preparation of the chitosan solution, 90, 50, and 10%. These results demonstrate the presence of the microneedles uniformly organized on the patch surface. In the case of the chemical composition, the characterization of the chemical composition by FTIR showed that the chitosan and the chitosan meloxicam microneedle patches preserve the chemical composition of the polymer and drug. Here we can see the spectrum of pure chitosan. These are the spectrum for the patches by varying the concentration of the acetic acid, 10, 50, and 90%. Here we can see while increasing the concentration of the acetic acid, the absorption of the acetic acid are more predominant. Therefore, it is better to work with a low concentration of acetic acid, 10%. Here we can see that the meloxicam spectrum, which has these predominant bands, and this is observed the spectrum for the patch with meloxicam. We can appreciate more this peak when working with the lowest concentration of the acetic acid. For the mechanical testing, reported that the higher acetic acid percentages reflected greater resistance to compressing force as temperature increased. So figure A present patches at 50% of acetic acid and figure B at 10% of acetic acid. If we compare the results at 2 Celsius degree, we will notably see that the microneedle patches prepared at 50% of the acetic acid reflect greater resistance to compressing compared with the microneedle patches prepared at 10% of the acetic acid. The in vitro microneedle insertion testing demonstrated that the microneedle patches were able to penetrate the skin of the cow's ear. So here we can see how the entire microneedle patch looks. The other figures show that the penetration on the skin was successful. So as a conclusion, we have the chitosan meloxicam patches presented an organized distribution of an homogeneous dimension of microneedles. The chemical composition of the chitosan and meloxicam were successfully preserved when using acetic acid as the solvent at 10%. The microneedle patch was able to penetrate the skin. So in general, we can say that our chitosan meloxicam microneedle patches might be a suitable way to manage pain in cattle after routine procedures, solving the problem of controlling the drug administration and avoiding multiple injections or oral administration per day. As a future work, we would like to include the study of increasing the amount of drug to be contained per patch and achieve the reduction of using several patches per cattle. In addition, in vivo experiment cattle will be developed to evaluate the transdermal drug delivery and pain relief during routine management practices. And I have to point out that at this point of the project, we have already made some of these work. We have already increased the amount of the drug. We already did some in vivo experiment, But however, based on the result that we already got from the in vitro study and the in vivo study, we still need to optimize the drug release of the meloxicam. So we would like also, again, to perform some in vivo experiment. So mainly I'm working on that and optimizing the drug release. So far, I think we are having good advances on this part of the project. So we'll see after we perform the MIBA study how that goes. So as an acknowledgement, this project has been found by Chancellor Fund for Innovation and Collaboration of the University of Arkansas, and this is my wonderful team in the Almodovar's lab. Now I'll be happy to answer any questions that you have. Thank you.
All right, thank you. That was a fantastic talk. So we will just jump straight into the questions. And again, for the chat, you're welcome to keep asking questions as we go. Certainly don't. This isn't a cutoff for that. First question is: are there any potential applications for microneedle drug delivery for humans?
Sorry, can you repeat the last part?
Oh yeah, sure. Are there any potential applications for the same type of microneedle drug delivery in people?
Oh, okay. Well, there are already currently some study where they are doing some patches for the transdermal delivery of insulin to treat diabetes.
Oh.
So yes, currently they are working on that, and there are another type of drugs that I can't remember, but mainly that one, yes, is a new alternative that they are working right now. So less painful, less stressful, even for animals, for people. So yes. So
interesting. Okay, very cool. Let's see. How do you control the rate of release of the medicine?
The rate of release.
Yeah, you kind of. It sounded like maybe that's something that you're going to look more at, look more closely at in some of your future work. You said you want to optimize it, but just in general, how do you try and control that rate of release?
Well, we did some study like in vitro release analysis HPLC. In order to get some percentage of the accumulation drug release profile of the meloxicam, and we did that for different amount of time, like certain time points from 1 hour to even 7 days.
Oh, wow.
And we did the same thing in animals, but based on the data that we got, we still need to work on that since we didn't get like more than the 50% of the drug side of the animal body and even in vitro after 7 days. So we tried that the first time. So yes, that's mainly how we are getting those rates right now.
Okay. So if you want to, I mean, I know you're still working on it, but kind of looking ahead, what kind of things, what sort of things do you want to try to make it? So I think you said you only released about 50% of the medicine, right? So
yes.
what kind of things would you what sort of things do you want to try and do to try and increase that amount of medicine released?
Okay, maybe the problem is that meloxicam is a type of drug that is quite not easy to solve as a solvent. It needs a solvent.
Okay.
So we try with the chitosan, but it's still a little bit harder. So we might need to add adding new polymers to the solution. So that could be a new recipe that will help the drug to get inside, like to deliver the drug better than just with the chitosan. So probably adding a new polymer, maybe one or two that can even solve better than the chitosan with the meloxicam itself. So
okay.
Yeah.
Interesting. So you had 3 different acetic acid concentrations. What's the role of the acetic acid?
Of the acetic acid, in this case, just to propel the chitosan solution in this case.
Oh, okay.
So yes, to form mainly the hydrogel there, the solution.
Mm-hmm.
And the reason that we use the three different percentage of the acetic acid at the beginning,
Mm-hmm.
there was for two reasons. The first one was because we wanted to evaluate if we can fabricate the microneedles.
Mm-hmm.
So we select three different concentrations that can dissolve the chitosan and see using which one can prepare a better one. Like the second reason was
Sure.
just trying to identify if we can modulate the mechanical properties of the microneedles.
Okay.
This is because the microneedle patch must be able to penetrate the skin over the
Mm-hmm.
dermis.
Mm-hmm.
So yeah, it was that.
Okay. Interesting. Yeah, the microscopy images were outstanding. That was really. I personally love seeing that kind of, where you have like the repeated structures going in all directions.
Yes, exactly.
So with what other kind of medicines, or I guess what's the, trying to think of the right way to ask this. So I'm assuming that you could do something like that, like you talked about in people, they've got these, or they're trying these with insulin. What other types of medicines do you see this technology being used for?
Oh, well, even vaccine antibiotics already.
Mm.
Different type of antibiotics. I have read like they've been using different, not just for dissolving microneedles. It could be the different other type of microneedle that I mentioned at the beginning.
Mm-hmm.
They're trying with antibiotics and insulin,
Mm-hmm.
and even they are trying to get some to treat cancer. But
Really?
yes. So it's kind of interesting. Yeah, I didn't know about that, but they are trying to use that like mainly for the vaccine part.
Uh-huh.
Yeah, yeah.
That's really cool. All right. Um, are there so with this patch? Does the patch stick well? I know that's a weird question, but like if on the cows, like if they're moving around, does it stay in pretty well?
Actually, that's one main of the reason that they designed the animal department, because we are working together. They evaluate that part, like where exactly we can place the patch in the cows, and so
Mm-hmm.
they decide, like taking into consideration, like the best part was the ear. So it was not easy for the cow to take off the patch, and even they use some type of tape, adhesive band,
Oh,
to
okay.
stick, like to hold the patch in the cow's ear. Yeah.
Okay, that's really interesting. So is the use of the chitosan? I guess it seems like it's sort of a multi-purpose thing. It's pretty readily available, but you said it also has really good biocompatibility. Is that right?
Yes, that's right.
Okay. Are there any other materials I guess maybe not. I mean, certainly not exactly the same, but of similar interest that may have some different properties that could be used?
Oh, yes. As I mentioned, also we can use, for example, collagen,
Mm-hmm.
cellulose, uh, PVA.
Mm-hmm.
We can use PPA, I think. Those are the mainly one that I have read so far.
Okay.
The and gelatin, that is another quite famous if you want to use them as a solvent, and also as an alternative to deliver different type of drug with this technique. So
Yeah.
you make different type of combinations, different type of hydrogel, like even with alginate, hyaluronic acid, you
Oh.
can do quite interesting mixing of two different natural polymers, or even with synthetic ones.
Yeah.
Yeah, yeah.
Oh, that's really
So
fascinating.
for us, it was mainly based on the antimicrobial properties
Mm-hmm.
that we decide with the chitosan. We tried with that one, but since based on the results, as I mentioned, the data that we got
Mm-hmm.
so far, we might need to another one to has a better degradation or release of the drug that will help to solve the
Okay.
patch. better.
Makes a lot of sense. Can you talk a little bit more about the design of the microneedle itself? I know you had the slide and you showed sort of the dimensions. Can you talk about, like if I guess basically like why those dimensions work well? I mean, certainly some of it has to do with how far it penetrates into the skin, but like the width, for instance, and the pitch, like what do those do? What are you trying to balance between those things?
Well, this was a decision that was made with my advisor and my previous grad student.
Mm-hmm.
But the way that they decide is also based on the dosage, the amount of drug that they really want to try there.
Oh,
So,
okay.
and yeah, so the dimension were set it based on that and try to see how that works. But at this point, we might need also, we are considering fixing that or increasing sizes since we really want to have um, to increase the dosage of the drugs itself.
Mm-hmm.
And they didn't want to try like a higher one because there are some standard dimensions that you need to use for microneedles to not cause any pain or it's not harmful for skin.
Okay.
So those dimensions has to be taken into account. So they try with these ones first,
Mm-hmm.
but we need to just to make some improvement, just to add more drug. Yeah.
Okay. And can you go to the part where you had the steps to actually make the patch itself?
Yes, this one.
Yeah, yeah, yeah. So it looks like the total time is about 3 days or so to make it. Is that right?
Yes. With this part of the project,
Mm-hmm.
yes. It was quite like 3 days, but we made some advantages that we can make all of them like a whole day, like 24 hours. So there are
Okay.
some of the improvement that we have already made to these patches.
Okay. Yeah, that's what I was going to ask. I was going to ask about like the potential for speeding that up, but yeah, if
Yeah,
you've already cut it down to a day, that's impressive.
yeah. Even with the solutions, with the centrifugation, even with everything, we are able to make like those the whole patches like in a whole day.
That's actually really impressive. I
Mhm.
mean, especially as I was, my first thought was, why not dry it at a little bit higher temperature? But I bet that would destroy the medicine.
Yeah.
So that's not a
Yeah.
great idea. All right. Oh, this is really fascinating. This is seriously cool work. So I'll give a chance for anybody to get any last minute questions in, but I think we can go ahead and start wrapping up a little bit. So I will say thank you again to you for a fantastic talk. It's super cool work. Um,
Thank you.
yeah, it's really. I mean,
It's been quite interesting actually. The first time I started reading about
mhm.
the new advances that they are doing in this field, it's quite impressive. So they are making different type of uh, it's a new alternative
Mhm.
that we didn't explore and had the opportunity to work on this.
Yeah.
Feel like, yeah, I really like this. It's kind of cool, even because it's related with my major. So yes.
Oh yeah, absolutely. Well, yeah. I mean, and it's clearly you're working on the cutting edge. So that I mean, that's really cool. That's
Yes,
five,
yes,
five, 5 years from now, this will be a normal
yes, it
thing. It's
is.
just going to be because of the work you're doing. So
Yeah.
that's super cool.
Yes.
It looks like that is all the questions we've got. So I will say thank you again. If I can get you to hang around for just 1 minute, we'll talk very briefly. Um,
No.
and
Yeah.
so, again, thank you for a wonderful talk. Thank you to everybody for coming out, asking great questions. Um, like I said, we'll be off the next 2 weeks, and we will be back on January 7th, I think. Let me double check that. Yep, January 7th with Dr. Andrew White from the University of Rochester. I hope you will all come back out for that. And in the meantime, have a great holiday. I hope you will get to take some time off and relax and recharge and be ready to come back for more research or industry work or whatever it is you're doing. And I hope you'll be back here on the 7th. So thank you all again to everybody for coming out, and be safe, get boosted, and see you next time. Thanks again. Bye.
Thank you.